All right, surgical technologists, let's get into Unit 5 where we talk about gastrointestinal organisms that are normal flora and pathogens. Let's get started. So the gastrointestinal environment is established pretty early in life. It remains relatively stable for your life, unless, of course, it is wiped out by antibiotics. Most bacteria are in the colon, and there's fewer amounts that are in the small intestine, and very few can survive the stomach, and that's because the stomach has a pH of about 1.0. It's very acidic. There's only a few that can survive the stomach. One such example is Shigella. There's a lot of flora in the gastrointestinal tract, including anaerobes, enterics, non-fermenters, staph, and streps. Anaerobes actually outnumber enterics 1,000 to 1. On this slide, we can compare the normal intestinal flora found in the upper small intestine, the lower small intestine, and also the large intestine. We can also see the numbers of organisms per mil. In the upper small intestine, we have about 10 to 1,000 organisms per mil, and those are usually Streptococcus, Lactobacillus, yeast, diphtheroids, which are Corynebacterium, those are the gram-positive rods, and Enterococcus faecalis. In the lower small intestine, we have many more organisms at about 1 million to 10 million organisms per mil, and those are usually enterics and Bacteroides, which is an anaerobe. And then finally, in the large intestine, here we have the most organisms. We have about 100 billion organisms per mil, and that would consist of anaerobes, enterics, staphylococcus, and pseudomonads. Here I'd like to talk about some ways that we can actually avoid colonization with pathogens. So one way we can avoid getting sick with pathogens is the competition of pathogens for nutrients. So normal flora and pathogens compete for nutrients. Also, the acidity of the stomach protects us from getting anything pathogenic. The pH in the stomach is far too hostile for most organisms to survive. Like I said in the previous slide, some organisms can survive it, like Shigella species. Also, normal peristalsis of the intestine. The small intestine is constantly in motion, and that pushes organisms that are potential pathogens out. We have a mucus layer that lines the gastrointestinal tract, and that traps organisms and or helps them pass through the gastrointestinal tract. And then finally, we have secretory IgA, which is an antibody present in mucosal secretions like saliva, tears, and the gastrointestinal tract. And then we have phagocytic cells. These are all ways that the host is protected from getting colonized by pathogens. The clinical symptoms of a gastrointestinal infection are actually many. The most common presenting symptom is diarrhea, but again, it really depends on what type of gastrointestinal infection you have. It could be bacterial, or it could be parasitic, or it could be viral. Abdominal cramping is another symptom associated with gastrointestinal infections. Dysentery, which is basically a term that means severe diarrhea associated with cramping in blood cells in the stool. You may have a fever and you may be dehydrated. Other symptoms that are common with gastrointestinal infections would be things like nausea and vomiting. There are a number of different agents that can cause gastrointestinal illness. And the ones that we're going to focus on are basically on this list. We've got bacterial agents of infection, viral agents of infection, and parasitic agents of infection. So some of the bacterial causes of gastroenteritis would be things like Shigella, Salmonella, E. coli 0157H7. That's a different strain of E. coli that is normal flora of the gut. Campylobacter species, Vibrio cholera or Parahemolyticus, Clostridioides difficile, Clostridium perfringens, Bacillus cereus, Staphylococcus aureus, and then a few other ones we could add on here would be Yersinia intercolitica and also Aeromonas hydrophilia. Some viral causes of gastroenteritis would be norovirus, rotavirus, and astrovirus. And then some parasitic causes of tummy troubles would be Giardia lamblia, Cryptosporidium, and Isospora, and that can happen in especially in immunosuppressed people. And then finally, Entamoeba histolytica, which is a parasite that causes bloody diarrhea. So there are a number of reasons why a person may have gastroenteritis. So you may be asking yourself, would a gram stain be helpful on a stool culture? And for the most part, gram stains are not particularly helpful when looking at stool samples. They're generally not performed. They might be helpful if we are looking for white cells. If you're looking for polymorphonuclear cells, you could do a stool WBC or you could do a gram stain to determine what kind of white cells are in the sample, if any. And it may also be useful when evaluating an overgrowth of Staphylococcus or yeast. So if you're not seeing the other flora that is usually in a stool sample, you might see Staphylococcus or yeast taking over. And then if you're looking for a specific morphology, such as the gull-wing shape of Campylobacter or a large gram-positive rod, something like Bacillus with spores, or a Clostridium perfringens where you see a boxcar-shaped gram-positive rod, in these cases, it might be useful to use a gram stain as well. But for the most part, gram stains aren't helpful because you end up seeing a bunch of mixed flora and nothing really jumps out at you most of the time. As you can imagine, it's virtually impossible to screen all symptomatic patients for all gastrointestinal pathogens. It's actually helpful to get a good patient history so we can start to narrow down what exactly the etiology of the gastrointestinal illness is. For example, what's the patient's age and immune status? Are they very young? Are they very old? Are they immunosuppressed? Have they traveled recently and to where? Is it possible they have cholera? Is it possible they have giardia from traveling? What types of food have they consumed recently? Have they consumed fried rice, sushi, or oysters? These are all things that you should be thinking about when we're trying to narrow down what organisms to culture for. When getting a good patient history, and you're trying to figure out the possible age of infection, here are some questions that you should be asking your patient. Did you drink from a mountain stream lately? Is it possible you have Giardia lamblia? Have you ingested dairy products that seem to be off or bad? It could be you have salmonella or listeria. Have you traveled to the east or Gulf Coast? There people like to eat their raw oysters. You might have a case of Vibrio. Have you ingested eggs that are spoiled or potato salad that's spoiled? You should be thinking things like Staphylococcus aureus and its toxins, or possibly salmonella. Have you ingested buffet fried rice? Fried rice is a great culture media for things like Staphylococcus aureus or Bacillus cereus. Have you ingested undercooked hamburger? E. coli 0157H7 is associated with undercooked hamburger. You may remember the story of the Jack in the Box restaurant where the child ate undercooked hamburger and ended up getting hemolytic uremic syndrome. That was many years ago, but it was associated with E. coli 0157H7. Have you ingested shellfish lately? If you've ingested shellfish that was not cooked properly or contaminated, you could have parasites or Vibrio parahemolyticus. Or are you a patient that is immunosuppressed? And there could be a number of things causing your illness, including things like Cryptosporidium or Isospora. If you have a bacterial gastroenteritis, it could be caused from a toxin-producing strain of bacteria, or it could be caused from a bacterial strain that eats away at the intestinal mucosa. Here I want to talk a little bit about enterotoxins. An enterotoxin is a substance that stimulates hypersecretion of fluid and electrolytes, which overwhelms the absorptive capacity of the large intestine, and you have diarrhea. Usually, you have excessive diarrhea, and the diarrhea does not have WBCs or RBCs present. The organ involved in enterotoxins that is affected is the small intestine. Some organisms that are enterotoxin-producing strains would be things like Staphylococcus aureus, Bacillus cereus, Cryptosporidium, Clostridium botulinum, and Vibrio cholera. Other organisms are said to be invasive pathogens. Okay, here we have organisms that damage the mucosa of the large intestine, resulting in the loss of white cells, blood, and protein. Here we do have white cells and red blood cells that are seen in the stool. Inflammation occurs and occasionally invasion of the bloodstream. The organ involved when pathogens invade is the large intestine. Here we have organisms like Yersinia intercolitica, Campylobacter species, Shigella, Salmonella species, and Entamoeba histolytica. These are all organisms that invade the intestinal mucosa. You could even go as far as saying that we have large bowel infections and small bowel infections. A large bowel infection is one that is said to be an invasive infection. So here we have small volumes of diarrhea, however, there are red cells and white cells present. Usually your patient has abdominal pain, fever, and just a mild dehydration. And there's a slow incubation period for this type of disease. Usually the incubation period is about 1 to 3 days. Okay. We have small bowel infections, and usually those are more associated with bugs that produce the enterotoxins. Here we have large volumes of diarrhea without the presence of red cells or white cells. They may have little to no abdominal pain. Your patient is probably not going to have a fever. However, they do have severe dehydration because of the loss of fluids. This is a rapid incubation period at 6 to 12 hours, and usually the symptoms are from the enterotoxins of the organism more so than the organism itself. So infections of the large bowel are usually associated with Salmonella, Shigella, Yersinia, Campylobacter, C. difficile, and hemorrhagic E. coli. Also, some other ones you can put on that list that are invasive would be Shigella and Entamoeba histolytica. Then we also have organisms that are small bowel pathogens. These would be things like Staphylococcus aureus, Vibrio cholera, and then other strains of E. coli that are not the 0157H7 strain. Other organisms that we can put on this list would be Bacillus cereus and Cryptosporidium. So what happens when you have a gastrointestinal illness? What's the outcome of that? Are you going to get better? Are you going to get worse? What can happen? Okay. Usually they're self-limiting. Most of the time, the physician is not going to treat the patient with antibiotics. Okay. Now, Shigella, usually the patient does need antibiotics, but all of your other gastrointestinal illnesses, the doctor's usually going to let it run its course. The reason why is we don't want to turn the host into a carrier of that disease, and that's especially true with Salmonella. One other thing that can happen is severe dehydration. That may be a consequence, especially when you have an organism that's producing an enterotoxin. Your patient could develop toxic megacolon. This is when the colon swells and holes start to develop in the colon, and then stool eventually spills out into the abdominal cavity, which of course is not good. Your patient may develop Reiter's syndrome. This is a reactive type of arthritis. Your patient may develop hemolytic uremic syndrome. That's associated with E. coli 0157H7. And then your patient may develop a malabsorption or absorptive surface of the colon that is damaged, and that's because some of the colon has been sloughed away. So these are all outcomes of gastrointestinal illnesses, and this is why we do want to take them seriously. Most of the time, if you're a strong, healthy adult, the physicians are going to let the disease take its course and they're not going to treat you with antibiotics. Now, that's most of the time. However, there are circumstances that would lead the doctor to prescribe antibiotics. And some circumstances of that would be if the patient were immunosuppressed, if the patient has recently traveled to a developing country, if the patient is experiencing bloody stools, if they've had diarrhea for an extended period of time, greater than 3 days. Sometimes the disease requires intravenous rehydration with IV fluids. And if the patient has a fever that won't resolve, sometimes they'll treat with antibiotics. That especially is true in elderly people, immunosuppressed people, and very young people. Let's go ahead now and take a look at some of the organisms that cause gastrointestinal problems. Okay, the first one I want to talk about is Shigella species. It is an invasive type of bacteria. You will see red cells and white cells in the sample, and they produce what's known as shigatoxins. These are toxins produced by Shigella. Okay, It is a very serious disease, especially in children, where the neurotoxins can be so potent that it can prevent the anal opening from closing in young adults, and they can actually lose tremendous amounts of fluids. Okay? The symptoms usually are 24 to 48 hours after you've ingested the organism that causes you to be sick. So again, Shigella usually comes from contaminated food or water. It's highly transmissible, and humans are the only natural host for this organism. So if you catch it, you're actually catching it from another human or human sample like feces. Um, it is reportable, and what I mean by that is this has to be reported to the state health department. And there's 4 common species. I'll talk about those in the next slide, but Shigella sonii is the most common in the U.S., while Shigella dysenteriae is the most virulent. This causes a very typical type of dysentery, and the best strategy for isolation is to put it on blood agar, McConkey or EMB, hectoenteric or XLD in a broth. It's one of the few bacterial causes of gastroenteritis that may be treated with antibiotics like ampicillin or tetracycline. So Shigella and Salmonella and even Campylobacter and E. coli 0157H7, they can all be serologically typed. So Shigella species, they have a scientific name, and then which is a genus and species name, and then they also have a serological typing name. So Shigella dysenteriae, that's the most serious type of Shigella. It's not seen in this country too often, but this is known as Group A Shigella. We've got Shigella flexneri, which is known as Group B Shigella, Shigella boydii, which is Group C Shigella, and then the most common Shigella seen in the United States is Shigella sonii, which is known as Group D Shigella. This brings us to Salmonella, and most people have heard of Salmonella. This is a gastrointestinal organism where the symptoms appear in 1 to 3 days. It usually is associated with eating raw meat, poultry, milk, and eggs. It's most commonly you're most commonly infected by eating something contaminated with animal feces or human feces. Other modes of transmission are possible also. For instance, person-to-person transmission is common. You can actually get it from another person, usually because those people aren't washing their hands appropriately. It is communicable and reportable to the state health department, and it may cause other bad things, including enteric fever, also known as typhoid fever, septicemia, or a person can just be a carrier. That is one reason why they don't treat Salmonella patients with antibiotics, because it could actually prolong the illness. Salmonella can actually survive phagocytosis, go into the bloodstream, the gallbladder, and into the gallbladder and get stuck in the gallbladder as a glob, and then is constantly being shed into the stool. That's what happens when a person is in the carrier state. Salmonella has a very difficult type of nomenclature. There's actually 2 species, Salmonella enterica and Salmonella bongeri. Enterica has 6 subspecies and bongeri has 1 subspecies. They're all divided into serogroups based on their somatic antigens, which are the antigens attached to the cell body, and they can be further divided into 2,300 other serotypes based on their antigens on the flagella. The CDC recommends that the organism be identified as Salmonella in serotype, omitting the species name. For example, we would call Salmonella typhimurium Salmonella serotype typhimurium group B. Group B refers to the somatic antigen classification. This is probably all more than you need to know, but I just wanted to mention that Salmonella nomenclature can be pretty extensive. I just want you to understand that when we call Salmonella Salmonella typhimurium, we're actually referring to a serotype and not a species name. Actually, they all end up getting treated the same way, so it doesn't really matter to the microbiologist. However, it does matter to the epidemiologist. If you have Campylobacter, you usually develop symptoms 2 to 5 days after the exposure of bad meat, poultry, milk, and animal feces. If you were to stain Campylobacter, it has a very unique gram stain appearance. It actually has a gull-winged shaped gram-negative rod. Okay. It may require a prolonged counterstain with safranin because it doesn't seem to take out the counterstain very well. It is not fermentive or oxidative, and it occurs as a gastrointestinal pathogen 7 times more frequently than Salmonella or Shigella. Oftentimes you have Campylobacter and you never even knew it or heard about it because you're usually better by the time you decide to go to the doctor. Some symptoms of Campylobacter would be cramping, abdominal pain, bloody diarrhea, chills, and fever. There are a couple species of Campylobacter, but usually the one that causes the most trouble for humans is Campylobacter jejuni. But there's also species Fetus intestinalis, Fetus jejuni, Campylobacter coli, and Campylobacter laureus. All of these cause gastroenteritis, especially Campylobacter jejuni. It is a microaerophilic organism, and what I mean by that is it requires 85% nitrogen, about 5% oxygen, and 3 to 10% CO2. The optimal growth temperature for Campylobacter is 42 degrees, and it is a slow grower. It does require up to about 48 to 72 hours of incubation. There is special media for growing Campylobacter, including Campylobacter blood agar and CVA media, which contains vancomycin and other inhibiting substances to prevent the growth of most other organisms. Gram-positives and gram-negatives are inhibited on CVA and Campy blood agar. Escherichia coli usually is not a bad little dude. Many strains of Escherichia coli are completely normal flora of the gut. However, there can be some toxic categories of Escherichia coli, including the enterohemorrhagic, which are the EHECs, or the enterotoxigenics, which are the ETECs. You probably heard of hemolytic uremic syndrome, and that is caused by a strain of E. coli that is an enterohemorrhagic strain. This includes the strain 0157H7, and it produces Shiga toxins. Now, Shiga toxins are toxins that are Shigella-like, and that's why they're called Shiga toxins, but they are not necessarily made from Shigella, just so you understand that. They may cause hemorrhagic colitis, okay? We also have the enterotoxigenic ETEC E. coli's, which are mostly associated with traveler's diarrhea, where you have large volumes of diarrhea. It's similar to cholera in total volumes. Usually they lack a fever, they have nausea and abdominal cramping. Miscellaneous toxins are produced with enterotoxigenic E. coli. Adults and children are both affected, and it's typically not invasive. E. coli 0157H7 is a specific Shiga toxin producing strain of the EHEC organisms. It is an organism that will not ferment sorbitol, and that becomes important because there's a selective media called McConkey with sorbitol, and it is selective for your enterics, so either E. coli will grow in there, but some are sorbitol fermenters and some are not. And it's your sorbitol non-fermenters that we are concerned might be E. coli 0157H7. Once sorbitol negative strain is isolated, the organism can then be serotyped with the 0157 and the H7 antisera. This is an agglutination test with specific antisera for the strain of E. coli. Like I've said before, E. coli can be totally normal in the gut. However, there are Shiga toxin producing strains that can cause all kinds of gastrointestinal problems. E. coli 0157H7 produces a couple different potent toxins, including virotoxin 1 and 2 and enterohemolysin. They're lactose positive but sorbitol negative, and the media of choice is McConkey with sorbitol. Again, we're looking for sorbitol non-fermenting organisms to see if they are E. coli, and then if they're E. coli, we can serotype for 0157H7 antigen. This organism can lead to what's known as hemolytic uremic syndrome, where toxins from the Escherichia coli go into the bladder and destroy red cells. The kidneys fail trying to filter out shards of broken red cells. Usually with hemolytic uremic syndrome, the patient has a low-grade fever, no WBC seen in the stool, but RBCs can be seen leading to hemolytic anemia. They have low platelet counts and kidney failure. It's been estimated that the strain can be found in about 1 to 2.5% of beef samples. There is a human vaccine that's in progress for 0157H7. However, it has not been made yet at this point, but there is a vaccine for cattle that already exists. When I think of Listeria monocytogenes, I think of the Blue Bunny ice cream scare that happened in the early 1990s. The ice cream actually was contaminated with Listeria, and that makes sense because Listeria is an aerobic gram-positive rod that tolerates temperatures at 4 degrees Celsius. And it does survive at 35 degrees Celsius too. And so in the microbiology lab, a lot of the times what we'll do is we'll start to culture it at 4 degrees in the refrigerator, and then once a week we'll sub it to body temperature plates, which is 35 degrees. The thing about Listeria monocytogenes is that even though it's a gram-positive rod, it looks a lot like a Streptococcus group B on the plate. So it kind of has a soft zone of beta hemolysis. And then it has an unusual umbrella motility pattern. I'm going to show you a picture of this in the next slide. So here on the left, we can see a gram stain of Listeria monocytogenes. They are said to be gram-positive rods that look like they're laying in log jams. You don't actually see the picket fences or palisading effect like you would with diphtheroids. This is definitely a log jam formation. And then in the image on the right, we've got a sulfide indole motility media, and we're looking at motility, and you can see that parasol or umbrella-shaped motility in the tube. What happens is the organism grows up the tube where you stabbed it, and then it grows along the top, and then it falls. And that's why it ends up making this umbrella or parasol-shaped motility pattern. Helobacteriosis is a condition caused by the organism Helicobacter pylori. It's an infection of the gastrointestinal tract. Symptoms of helicobacteria would be things like inflammation, gastritis, ulcers, and stomach injury, including stomach cancer. It causes inflammation of the stomach lining. It's a gram-negative spirochete and produces urease enzyme and cytotoxin VAC-A protein. The VAC-A protein is a protein that is a cytotoxin and forms anion channels in target membranes, destroying cellular mitochondria. Children living in unsanitary or overcrowded conditions are more prone to helicobacteriosis. The diagnosis is usually made by endoscopy or the breath test for urea. Treatment is usually amoxicillin, clarithromycin, metronidazole, or tetracycline. Yersinia intercolitica causes an infection similar to Salmonella. We don't see it too much in the United States. We usually see it more commonly in Europe or northern Canada because it prefers cooler climates. It's actually known to contaminate blood products too because it can tolerate refrigeration. This organism, although it can be a GI pathogen, is not routinely screened for. It is a member of the enteric family, the Enterobacteriaceae, and it's not Yersinia pestis. Don't confuse this with the organism that causes the plague, which is Yersinia pestis. Humans are exposed via contaminated food or water, and it is distributed widely in nature. The organisms are sturdy and they can tolerate refrigeration without the loss of organism recovery. And the optimal temperature to get it to grow is room temperature. That's unusual. Most enterics grow best at body temperature, which is 35 degrees, but this one does grow best at room temperature, which is 25 degrees Celsius. If we were trying to get this organism to grow or to culture this organism, we would put it on a selective media known as CIN agar. This is selective for Yersinia intercolitica. It's not going to grow anything but that. And the organism has an interesting macroscopic appearance. It actually looks like a bullseye on CIN. So it has a pink periphery and then a clear center and then a pink dot right in the middle. The new name for Clostridium difficile is Clostridioides difficile. It can actually cause pseudomembranous colitis or antibiotic-associated diarrhea and enterocolitis. It is normal in the soil and water and also the intestinal tract of some people. It's a gram-positive rod with subterminal spores. That means the spores are not on the end. They're actually in the middle of the cell body. And microscopically, they are gram-positive rods that may or may not show these subterminal spores. Usually we detect Clostridium difficile by looking at the toxins A and B. Toxin A is associated with fluid accumulation and the excessive amounts of fluids being lost from infection. And toxin B has to do with tissue destruction. So you're going to end up with white cells and sometimes red cells in the stool because of tissue destruction. There is a selective media for Clostridium difficile or Clostridioides difficile, and that's called CCFA media, cycloserine sulfoxetine fructose agar. But it is best to identify Clostridioides difficile by the ELISA test. That's a serological test. We are looking to see if the patient produces the toxins. Clostridioides difficile is difficult to culture. Like I said, you could culture on CCFA media or an anaerobic PEA. If you remember PEA, this is a selective media for gram-positive organisms. You could do that, but it is difficult to culture. It's best to help diagnose this by ELISA test. However, if you wanted to speciate Clostridioides, you would put it on what's known as an egg yolk agar and look to see if the organism produces the lipase or lecithinase enzyme. If your organism produces lipase, what you'll see is an oily appearance on the colonies. And if your organism produces lecithinase, you will see a precipitate in the media around the organism. Here's an image of egg yolk agar, and this is what we use to speciate our Clostridium and also Clostridioides. As you can see in the top image there, we have the colony of the organism growing on the media, and then there's a precipitate that grows around the organism inside the media. This is because the organism is a lecithinase enzyme producer. On the bottom image, you can see that the colonies appear to be oily or have the mother of pearl sheen, and that's because your organism is a lipase producer. That's a fat-reducing enzyme. Vibrio cholera is the next organism that I want to talk about, and it does produce what's known as cholera. And like I mentioned in earlier PowerPoints, there is a current pandemic of Vibrio cholera taking place in the world that's been ongoing since 1961. Several 100,000 deaths have occurred from cholera in Africa, India, and basically it has affected 2.9 million people every year. Vibrio cholera produces a toxin, and this toxin has been the cause of 7 pandemics, and we are now currently in the 8th pandemic. The pandemic currently includes Chile, Peru, Colombia, Brazil, Africa, and India. There's also a Haitian pandemic that occurred in 2010 where there were 60,000 cases of Vibrio cholera. Vibrio cholera causes the most serious, severe diarrheal disease. Shock and death can occur in 24 hours from fluid loss and cardiovascular collapse. Gallons of diarrhea is lost every day. It is characterized by vomiting on what's known as rice water stools, and basically the stool looks like rice water because of flecks of mucus and tissue in the stool. Patients can go into shock within 12 hours of initial symptoms, and they are infected from contact with contaminated water and food, and usually that's seafood. If we wanted to culture Vibrio cholera, we could do so on TCBS, thiosulfate citrate bile salts agar, which differentiates sucrose positive fermenting from sucrose negative fermenting strains of Vibrio. Usually it is Vibrio cholera that ferments sucrose. Okay? We are not specifically culturing for Vibrio cholera unless the doctor is actually looking for this organism, but it will always be beta hemolytic and oxidase positive. Antibiotics generally are not given for this disease, just replacement fluids and glucose solutions. Cholera can be identified in a number of different ways. One way we can identify Vibrio cholera is by biochemical and enzymatic testing, or where we're looking at the metabolic activities of the organism. One way that we can identify it is by using an analytical profile index strip that will actually code out the organism Vibrio cholera. We can do what's known as the Vibrio string test, where we emulsify the colony in 0.5% deoxycholate, touch it with a loop, and move it away from the slide. If there's a mucus-like string that appears the organism and the slide, that is a positive test for the Vibrio string test. And then finally, like most gastrointestinal pathogens, we can serologically type this organism. Aeromonas hydrophilia can cause gastrointestinal symptoms as well as wound infections associated with fresh water. This is a gram-negative rod. It's beta hemolytic on blood agar, and it is cytochrome oxidase and catalase positive. It will reduce nitrates to nitrites. There's strong evidence that suggests it's an agent of diarrheal disease, although that is frequently encountered. What we look for is beta hemolytic organisms on blood agar that are cytochrome oxidase positive, and then we can metabolically identify them. Also, Aeromonas hydrophilia will produce a toxin known as the asaotoxin, which was named after the person who discovered it. This is an alpha hemolytic toxin that destroys red cells. Some wounds that are treated with medical leeches have been infected with Aeromonas since they were colonizing those leeches. As you can see, Vibrio cholera and Aeromonas hydrophilia are similar metabolically. They both are beta hemolytic and cytochrome oxidase positive, but as you can see, Vibrio cholera is escalin negative and VP negative and ornithine decarboxylase positive, whereas Aeromonas is escalin positive, Voges proskauer positive, and ornithine decarboxylase negative. Lactobacillus species is a chaining gram-positive rod, and you can find aerobic and anaerobic species of Lactobacillus. 90% of the time, this is normal flora, and I think of it as normal flora of the gut, the respiratory tract, and especially the female genital tract. It's lactobacillus that keeps the vagina at a pH of 4.5 due to the production of lactic acid. It actually prevents females from getting yeast infections and other genital tract infections. Lactobacillus can be an alpha hemolytic colony, much like a lot of streptococcus can look on sheep's blood agar. However, lactobacillus has been known to cause ulcerative colitis via bacteremia and the collagen-binding protein. It has a wide range of fermentative qualities, including fermentation of yogurt, wine, and other fermented foods. When I think of Streptococcus mutans, I think of an organism that is included in a biofilm in the mouth. It's Streptococcus mutans that usually causes tooth decay and dental disease. But it is an aerobic gram-positive cocci that can cause bacteremia, endocarditis, and sometimes gastrointestinal tract illnesses. There are a number of ways to identify gastrointestinal pathogens. Most of the time, we're going to identify them in the microbiology lab using metabolic properties. In other words, do they ferment certain sugars? What are their byproducts of glucose metabolism and things like that? Also, we want to know, will they tolerate high amounts of sodium chloride or salt? What are their fermentative properties? Will they ferment sucrose, lactose, or glucose? We can identify them using serological methods, and then we can also diagnose gastrointestinal pathogens using molecular methods such as polymerase chain reaction, where you take one strand or a few strands of genetic material and then amplify them, making several million copies of them. In this slide, I just wanted to show you a few examples of how we identify these types of organisms in the microbiology lab. So if you look at the image on the upper left, this is called an API, or an analytical profile index strip. And what you have there is a number of sugars and metabolic tests that are all kind of incorporated in this test strip. And what you do is you add a suspension of your organism to each one of those wells. And then whether or not the metabolism changes that chemical reaction will change the color of the well, or whether or not the organism will ferment the sugar will change the color of the well. And then we can actually come up with a 7-digit numerical code that's associated with that strip, and we can enter it into a database. And the database will actually tell us the name of the organism based on the metabolic and fermentative properties of the organism. If you take a look at the image on the bottom left, we can see that we have a type of media that has 3 sugars in it: lactose, sucrose, and glucose. And the colors that the tubes change are indicators as to which sugars are fermented by the organism. So in the first example, that tube that's all yellow throughout the tube is an example of where lactose and sucrose and also glucose are all fermented by the organism. And then we have instruments that can do measure metabolic activities of certain organisms as well. And if you look at the image on the top right, this is a machine called the Vitek, and the Vitek can actually look at several metabolic properties of an organism with a test card that looks almost like a credit card, and it has freeze-dried biochemicals in the test card. When we add the organism to the test card, the machine automatically reads the fermentative and metabolic properties of the organism on the machine. Or we can look for toxins. So for example, if we have a Clostridioides difficile, if you take a look at that cassette on the bottom right, this is an enzyme-linked immunosorbent assay test where we are looking for antibodies to the certain toxins. If your patient has the antibodies to the toxins, they're going to link up with a conjugate and form a colored line. Just to summarize, there are a number of bacterial pathogens of the gastrointestinal system, there are a number of viral pathogens of the gastrointestinal system, and there are certainly a number of parasitic pathogens of the GI system. So here's a look at some of the bacteria that we talked about in this unit. We talked about Shigella and Salmonella. We also talked about E. coli 0157H7, that's the Shiga toxin producing strain of E. coli. Talked a little bit about Campylobacter jejuni, that's a common gram-negative spirochete that causes diarrhea 7 times more than Shigella or Salmonella does. We have Clostridioides difficile, that's associated with antimicrobial overuse. Wipes out your normal flora and causes pseudomembranous colitis. We have Helicobacter pylori, which can cause ulcers and other gastrointestinal problems. We've got Staphylococcus aureus that can produce toxins that cause food poisoning, such as toxins A and D. We've got Vibrio cholera that causes cholera. Aeromonas hydrophilia that can cause a gastrointestinal type of disease. And then finally, we've got Listeria monocytogenes. So all of these organisms are one that we covered in this unit today. And hopefully you found this unit to be pretty interesting. Bacterial causes of gastrointestinal problems are usually from foods that are risky. So some of the foods that are high risk for being contaminated with food toxins would be things like raw fish, sushi and seafood, eggs, sprouts, juices and ciders, milk, soft cheeses, hot dogs, deli meat. These are all foods that are high risk for bacterial contamination with toxins. How do I recover from a gastrointestinal organism? Drink plenty of fluids, stick to bland foods like toast, rehydrate, even if you have to possibly go to the hospital and get IV fluids, sometimes that's necessary. Rest, wash your hands frequently, that's the #1 best way you can prevent the spread of infection. And then finally, in some cases, you may be taking antibiotics, but that's usually not the case. All right, we covered a lot of ground in this unit. Hopefully you learned everything you ever wanted to know about gastrointestinal infections. If you have any questions, you know how to reach me. Have a great day.